Ever wonder how a massive oak tree can sprout from something as tiny as an acorn?
Or how about this, how a single fertilized egg can develop into, well, you,
It's all thanks to this incredible process.
of cell growth and division.
Yeah, and that's exactly what we're diving deep into today.
This microscopic world.
That makes life as we know it possible.
It really is mind-boggling when you think about it.
Like how?
Every living thing,
Every single one.
from the tiniest little bacteria,
To the biggest blue whale.
starts from just one single cell.
And that one single cell.
Has all the instructions it needs.
To build an entire organism.
It's like having a blueprint for an entire skyscraper.
Crammed into
A grain of sand
So how does that blueprint actually get used?
to make millions and billions of cells.
That's where the magic of the cell cycle comes in.
It's this super carefully organized series of events
That makes sure.
that a cell's DNA
And all of its components.
are copied accurately
Before it divides.
into two identical daughter cells,
Like a really carefully choreographed dance
where each step
happens in a specific order.
at a specific time.
Right on cue.
Okay, so let's break this cellular dance down.
Move by move.
What are the main phases?
Of the cell cycle.
Well, the cell cycle is divided.
Into two main phases.
Okay.
Interphase and the M phase.
And the M phase.
Is where the cell actually divides.
Mitosis.
That's the one.
So interphase is like all the behind-the-scenes work.
All the prep.
Getting everything ready.
For that big performance of mitosis.
Exactly.
During interphase, the cell is growing.
It's making proteins.
Doing its normal, everyday things.
Living its best life.
But most importantly,
it's
it's replicating its DNA
making a perfect copy.
of that whole genetic blueprint.
Yeah, I can imagine.
That's a super crucial step.
You wouldn't want to start building a skyscraper.
Without two copies of the blueprint.
Exactly.
And the cell takes duplication very seriously.
Like really seriously.
I mean, interphase takes up most of the cell cycle.
About 95%.
For a human cell.
Which shows you just how important this prep phase is.
Wow, so the actual dividing part is pretty quick then.
Well, compared to all that prep work
Yeah.
for your typical human cell.
Mitosis itself.
Only takes about an hour.
That's incredible.
It is.
Okay, so we've got interphase,
Where the cell is growing.
copying its DNA.
And then we've got mitosis,
The grand finale.
where the cell splits into two.
Can we dig a little deeper into interphase?
Sure.
What exactly happens during that phase?
So interphase,
It's got its own phases.
it has three.
3.
G1,
Okay, SD.
E2,
Okay, so G1,
stands for gap 1,
gap 1.
and it's a period of intense growth.
The cell is like bulking up.
Yeah, and metabolic activity.
Busy, busy, busy.
The cell is living its best life,
Single cell style.
carrying out its normal functions,
Getting bigger
getting ready.
for what's to come.
Then we get to the S phase,
Okay,
where the real action begins.
here we go.
This is where DNA replication takes place.
Those blueprints we were talking about.
This is where the cell makes an exact copy.
Of its whole genome.
The whole thing.
So the S stands for
Synthesizing. A new set of DNA.
Exactly.
During S phase, the amount of DNA doubles,
Okay.
but the chromosome number stays the same.
What does that mean?
So you basically end up with two identical copies
Of each chromosome.
attached at a point.
We'll call Hitty.
The centromere.
The centromere.
Imagine two identical volumes
Of an encyclopedia.
bound together.
I like that analogy.
It's a good one.
So the cell has duplicated its instruction manual.
And attach the two copies together.
What happens next?
Next up is G2,
G2,
or gap 2.
the last phase of interphase.
The final act.
During G2,
The cell keeps growing
cells will grow.
and producing proteins.
What kind of proteins?
The proteins that it needs for mitosis,
Okay.
but maybe even more importantly.
Well, more important than proteins.
This is where the cell double-checks its work.
Like a proofreader.
It scans the new DNA, looking for errors.
Making repairs where necessary.
It's like a really meticulous editor,
Going over a manuscript before it goes to print.
or looking for any typos.
Or errors in that genetic code.
It makes sense.
The cell wants to be extra careful
Yeah.
to make sure that DNA replication is accurate.
Because any errors in the code
Could have serious consequence.
for those daughter cells.
Right.
So once the cell has checked,
And double-checked its DNA
it's finally ready.
for the main event.
Mitosis.
That's right.
And mitosis is where all that prep work during interphase,
Pays off
it all culminates in this spectacular event.
of cell division.
All right, I'm ready to see this choreography in action.
Let's do it.
What are the key moves of mitosis?
So mitosis itself
Has more phases.
4 main stages.
4.
Prophase, metaphase, anaphase, and telophase.
Wow. Okay.
And each stage has distinct visual changes
You can actually see them.
Under a microscope.
It's like watching a carefully choreographed ballet.
Happening inside the cell?
No.
So let's start with prophase.
Okay.
What are the opening moves?
In prophase,
The chromosomes.
which were all spread out in the nucleus before,
Like a tangled mess of yarn.
start to condense
And they become visible.
as these distinct structures.
Like the cell is tidying up
Yeah, getting organized.
for that big divide.
You got it.
So we go from a blurry jumble.
To clearly defined chromosomes.
That's right.
And each chromosome
Now appears.
has two identical sister chromatids.
It's attached at the centromere.
Like those two volumes of the encyclopedia.
Bound together.
Precisely.
So the duplicated chromosomes are all organized.
Ready for action.
What else happens during prophase?
Another important structure starts forming.
What is it?
The mitotic spindle.
Okay.
It's made of microtubules,
Which are?
long protein fibers.
That act like ropes or cables
And it's going to be essential.
for separating the chromosomes.
Later,
So prophase is all about getting organized,
prepping for the main event
setting the stage for the separation.
of those sister chromatids.
Okay, what happens next?
Next comes metaphase.
Metaphase.
Maybe the most visually striking stage
Of mitosis?
of the whole thing.
Okay.
During metaphase, the chromosomes
They're condensed now.
attached to the spindle fibers
All lined up.
at the cell's equator.
I can picture it.
Continue.
A perfectly organized line of chromosomes.
Right down the middle of the cell,
Ready to be divided.
like they're waiting for a starting pistol.
That's a great analogy.
It really is like that.
This precise alignment is crucial
Absolutely.
to make sure each daughter cell,
Gets a complete set of chromosomes.
like dealing cards,
You want to make sure each player.
gets an equal hand.
Exactly.
It's amazing how the cell manages to do this.
With such precision.
Yeah, it really is.
It's an incredible process.
So the chromosomes are lined up at the equator.
Ready to go?
What happens next?
Anaphase.
This is it,
This is where the action happens.
the separation.
The centromeres that are holding those cysty chromatids together,
They split
and the spindle fibers pull the chromatids apart
like a tug of war.
in opposite directions.
Wow.
And it happens so fast.
And so precisely
Those encyclopedia volumes,
ripped apart at the binding.
each volume going to a different bookshelf.
What a powerful image.
It really is.
So we've gone from duplicated chromosome
Lined up at the equator,
to single chromosomes.
migrating to opposite poles of the cell.
What's the final stage?
The final stage is telophase,
Telophase.
where things start to wind down.
Okay, the chromosomes.
They've reached their poles.
They start to decondense
They're relaxing,
after their journey across the cell.
settling into their new homes.
At opposite ends of the cell,
What else happens during telophase?
a new nuclear envelope forms,
Around each set of chromosomes.
creating two new nuclei
So the genetic material is separated,
into two new nuclei.
but the cell itself is still one entity.
Right.
How does it complete the process of creating two daughter cells?
That's where cytokinesis comes in,
Cytokinesis. It's the process of dividing the cytoplasm,
which is.
the jelly-like stuff that fills the cell,
So it splits the cell into two
each with its own nucleus.
and a complete set of chromosomes.
Exactly.
So it's not enough to just duplicate and separate the DNA.
Nope.
The cell itself has to physically divide.
To complete the process?
And cytokinesis happens differently in animal and plant cells.
That's right.
So how does it differ?
In animal cells,
Okay.
cytokinesis involves a process.
That resembles
A drawstring bag.
a drawstring bag,
A furrow forms in the cell membrane,
Right.
gradually pinching inward
like tightening a belt around the middle of the cell.
until it splits in two.
And what about plant cells?
Plant cells.
They have those rigid cell walls.
So they can't just pinch themselves in half.
So how do they divide?
Instead of forming a furrow,
They build a new cell wall,
right down the middle of the cell,
like a partition.
dividing the cell into two compartments.
So even though cell division is similar across organisms,
The fine details can differ.
depending on the cell type.
That's right.
Fascinating.
Isn't it?
So we've covered mitosis,
One cell dividing into two identical daughter cells
essential for growth, repair, and replacement.
of cells in our bodies.
But there's another type of cell division,
We need to talk about.
meiosis.
That's the one.
This is where it gets really interesting.
I think so.
What makes meiosis so special?
And how's it different from mitosis? The main difference is that meiosis
Okay.
involves two rounds of division.
2 rounds.
Meiosis aura and meiosis the second.
But only 1 round of DNA replication?
That's right.
So you end up with 4 daughter cells
Each with half the number of chromosomes.
as the parent cell.
Exactly.
So in mitosis, we're making identical copies.
Yes.
But in meiosis,
We're creating cells with half the number of chromosomes.
why is that important?
That's crucial for sexual reproduction.
I see.
When a sperm cell fertilizes an egg cell,
Each gamete
contributes half of its chromosomes.
So the zygote has a full set.
Right.
It's like each parent.
Contributes half of the blueprint.
We're the new organism.
Exactly.
If meiosis didn't happen.
And each gamete had a full set of chromosomes.
Oh my God.
The number of chromosomes would double.
Every generation.
That would be chaos.
A recipe for disaster.
Yeah.
So meiosis ensures the continuity of a species.
By keeping the chromosome number consistent.
Are there any other important functions of meiosis?
Another crucial role.
Is increasing genetic diversity.
During meiosis,
Okay.
homologous chromosomes
From each parent.
pair up
And exchange genetic material.
through a process called crossing over,
It's like shuffling a deck of cards.
creating new combinations of genes
This genetic shuffling drives evolution,
by introducing variation.
making some individuals better suited to their environment.
It's an amazing process
It's a delicate balance.
between maintaining the integrity of genetic information
Through mitosis.
and introducing variation.
Through meiosis.
That balance is what has led to
The incredible diversity of life we see.
all around us.
Can you tell us more about those 2 rounds of division in meiosis?
Sure.
Let's start with meiosis,
Okay.
which is unique to meiosis.
Sets it apart from mitosis.
Okay.
Meiosis starts
After the parental chromosomes have replicated,
to make identical sister chromatids.
during the S phase.
Just like in mitosis.
Sounds similar so far.
But then things change.
How so?
Meiosis involves
The pairing of homologous chromosomes
and the exchange of genetic material.
crossing over.
Right.
This is where the shuffling happens?
Exactly.
These paired chromosomes
Or all them bivalents or tetrads.
line up at the cell's equator.
During metaphase,
And then during anaphase,
the homologous chromosomes separate.
moving to opposite poles.
Right.
So instead of sister chromatids separating
Like in mitosis.
in meiosis,
It's the homologous chromosomes that
get pulled apart.
That's an important difference.
Okay.
At the end of meiosis,
We have 2 daughter cells,
each with half the number of chromosomes as the parent cell.
but they're still duplicated.
Still 2 sister chromatids,
So we have the genetic material.
but still have 2 copies of each chromosome.
Okay, what about meiosis 2?
Meiosis the second is a lot like mitosis.
Like a mini mitosis.
You could say that.
The 2 daughter cells from meiosis
Enter meiosis 2.
without further DNA replication.
That's right.
During metaphase 2.
The chromosomes line up at the equator.
And this time,
It's the sister chromatids that separate.
during anaphase 2.
Yeah.
So meiosis the second is like
It's a cleanup mission
separating the sister chromatids.
that were still attached after meiosis.
And at the end,
4 daughter cells,
each with half the chromosomes.
as the original parent cell,
And each chromosome.
consists of a single chromatid. Now we've got,
Genetically unique cells.
thanks to crossing over,
It's an ingenious process.
that balances continuity of species.
And genetic diversity.
Meiosis is a great example.
Of nature's ingenuity.
Keeping life going and evolving.
This has been a fantastic overview.
Of cell growth and division.
We've gone from the cell cycle
To mitosis.
and then to the marvels of meiosis.
It's amazing.
How these microscopic processes drive everything.
Growth, development, the evolution of all living things.
Now that we've got the basics
We can shift gears.
and explore some of the implications of these processes.
For things like regeneration
Aging.
and diseases like cancer.
Sounds good to me.
I'm excited. What's really cool is how these processes
Cell growth and division.
are fundamental to everything about life.
Yeah?
From how a fertilized egg develops into a whole organism.
To how our bodies repair themselves throughout our lives.
These processes are always happening.
It's mind-boggling.
It is.
When you think about the sheer scale
Of cell division.
happening in our bodies every second.
Millions of cells.
Dividing constant.
Replacing old or damaged cells.
Keeping us alive and functioning.
And it's not just about replacing cells.
No.
Cell division is also what drives growth.
Like how we grow from babies to adults.
Exactly. Countless cell division.
Each one.
A meticulously choreographed ballet.
Of duplication and division. It's not just humans, though, right?
Right.
Plants need cell division to grow, too.
Absolutely.
Oh.
They have these special areas, meristems. They're at the tips of the roots and shoots,
Okay.
packed with cells,
That are constantly dividing.
allowing the plant to grow taller,
And spread its roots out.
further and further,
So from tiny bacteria,
to massive redwoods.
cell growth and division are essential.
For all living things.
It's what unites us all.
And it's the amazing accuracy of these processes,
The precision
of how cells copy their DNA and divide.
that ensures life continues.
It's pretty remarkable.
But it's not just about maintaining things as they are.
Right.
Meiosis shakes things up.
With its genetic shuffling.
That's what provides the material for evolution.
Exactly.
Meiosis is like nature's way of experimenting.
Trying out new combinations
Of genes.
that could give an organism an advantage.
In a changing environment.
It's all about adapting.
So we have this delicate balance
Between keeping the genetic information intact
through mitosis
and introducing variation.
through meiosis.
That's what has created
All the incredible diversity
of life that we see around us.
I'm curious, though. What happens when these processes go wrong?
When cell growth and division don't go as planned,
What can happen?
that's a really important question.
It is.
When errors happen during cell division,
Especially.
in DNA replication, the separation of chromosomes,
It can lead to,
uncontrolled cell growth,
which is a hallmark of.
cancer,
So cancer is basically a disease.
of runaway cell division,
The cells just keep dividing.
out of control.
They lose those checks and balances,
They regulate cell division
leading to tumors.
and the invasion of surrounding tissues.
It really highlights
The importance of that precise regulation.
of the cell cycle.
Absolutely.
If we want to understand cancer,
And how to treat it.
we need to unravel.
The mysteries of cell growth and division.
That's what's driving cancer research.
It's a fascinating field.
A bit sobering, too.
Yeah,
But it's not just about fighting diseases, right?
right.
Understanding cell growth and division
Opens up possibilities.
in other areas.
Like regenerative medicine.
Regrowing damaged tissues or organs.
Exactly.
Some animals can do that.
Like starfish.
And salamanders.
They can regenerate lost limbs.
Or even parts of their bodies.
It's incredible.
How is that even possible?
It all comes down to
Cell growth and division.
after an injury
These animals
can activate certain cells
that revert to a primitive state
capable of dividing rapidly.
and differentiating.
It's like they rewind their developmental clock
Wow.
and rebuild lost tissues from scratch.
It's like a built-in repair kit.
That would be amazing.
Imagine if humans could do that.
The potential is huge.
If we could understand.
And manipulate these mechanisms.
We might be able to regrow tissues and organs in humans.
That would revolutionize medicine.
It would be incredible.
It would.
So from everyday stuff like growth and repair,
To regeneration
cell growth and division,
are at the heart of life's resilience.
and adaptability,
Not just for individuals,
but for entire species,
driving evolution
shaping the diversity of life.
on our planet.
It's amazing
It really is.
that something as simple as a cell dividing,
Holds the key to so much
complexity and wonder.
in the natural world.
But we've talked a lot about cell division.
Yeah.
What about cell death?
That's important too.
It plays a role.
Absolutely.
While cell division is essential,
For growth and renewal,
programmed cell death is equally important.
we call it apoptosis.
Apoptosis, it's a regulated process.
Of cellular self-destruction.
When a cell is damaged or
Well,
infected,
just not needed anymore.
it gets signals
To self-destruct,
to start a cascade of events,
leading to its demise. It's like the cell gets a message.
saying, your time is up.
Time to bow out gracefully.
That's a good way to put it.
And this graceful exit is crucial for the health of the whole organism.
Why is that?
What happens if cells don't die when they should?
What happens?
If they don't undergo apoptosis? Those damaged or dysfunctional cells
Can accumulate,
and potentially lead to diseases.
like cancer.
Exactly.
So it's like a quality control mechanism,
Getting rid of bad cells
keeping the good ones.
to make sure everything functions properly.
It's fascinating
I think so.
that cell death is actually vital for life,
Apoptosis is
for
essential
the whole cycle of life,
from health and development.
to the evolution of all living things. It's amazing
It is.
how cell growth, division, and death
All work together.
to orchestrate this incredible symphony of life
A symphony that's constantly playing,
in every living thing.
from bacteria to whales.
It's a testament to nature's design
This has been a really enlightening deep dive.
and to cell growth and division,
We've gone from tiny cells,
to big concepts like evolution and aging,
and it's clear that these processes are fundamental.
to everything. And as we learn more about them,
We can find new ways.
to treat diseases, regenerate tissues, and maybe even,
Extend lifespan.
speaking of new discoveries,
I know some exciting stuff is happening.
in aging research.
Yeah, there are some interesting developments.
Can we talk about that?
Of course.
Okay, so aging is complex,
But one key factor,
is that cell division slows down.
as we get older,
Our cells can only divide so many times.
And over time,
that capacity diminishes
leading to the decline
in how our tissues function.
which we experience as aging,
So our cells have an expiration date,
in terms of how many times they can divide,
and scientists are trying to slow that down.
to maybe even extend lifespan.
That's a fascinating idea.
It is.
What are some promising areas of research?
One area that's really interesting,
Is
the study of telomeres,
telomeres.
those protective caps.
At the ends of our chromosomes.
They shorten every time a cell divides.
So they're like
The plastic tips on shoelaces,
to prevent fraying as they shorten,
they signal the cell to stop dividing.
which contributes to aging.
But some cells,
Like stem cells.
and cancer cells,
They produce an enzyme.
called telomerase,
Telomerase?
that can lengthen telomeres
So if we could activate telomerase,
in other cells.
could we slow down aging?
That's the idea.
It sounds too good to be true.
It's not that simple.
Why not?
Telomerase activity is tightly controlled.
For a reason.
Uncontrolled telomerase.
Is a hallmark of cancer. That makes sense.
We want to harness its potential.
Without causing uncontrolled cell growth.
It's a balancing act.
A tricky one.
But scientists are making progress.
Maybe one day we'll be able to,
Manipulate telomerase safely
and effectively.
to slow down aging.
That would be amazing.
It would be revolutionary.
This deep dive has really been eye-opening.
I'm glad.
It's made me realize
What?
the power and potential.
Of cell growth and division. It's
Yeah.
a field that's full of wonder and possibilities.
Speaking of possibilities,
What else are scientists exploring?
when it comes to cell growth and division,
There's so much to discover. One area that's super exciting is the study of stem cells.
stem cells,
Yeah.
those are the ones that can turn into
Different cell types.
like cellular chameleons.
Exactly.
They have so much potential.
For regenerative medicine,
This
they can
is-
become
Specific cell types
like heart muscle cells.
or neurons.
Right.
So we could use them to repair damaged tissues,
Instead of relying on organ transplants.
which are hard to come by.
And have rejection issues.
Exactly. So we could just use stem cells.
To replace those damaged tissues.
That's the goal.
That's what researchers are working towards.
It's like science fiction.
It really is.
But what are some of the challenges?
Of working with stem cells.
Yeah, what are they?
One of the biggest ones is
Saying sure.
that those stem cells
Differentiate.
into the right cell type.
And only that cell type.
You don't want a heart muscle cell.
Turning into a bone cell.
That would be a problem.
That's an understatement.
It's all about precision
Making sure they follow the instructions.
to become the cells we need.
Like programming a computer.
You need the right code.
To get the right output.
And researchers are getting better
At
at
understanding
signals,
that control stem cell fate.
which is leading to new possibilities.
For regenerative therapies.
Yeah.
So from telomere manipulation,
To stem cell engineering.
it seems like we're on the verge
Of some huge breakthroughs.
in cell biology.
Absolutely.
All thanks to our understanding.
Of cell growth and division.
This deep dive has been incredible.
It has.
It's shown me
Yeah,
these
and it's-
processes
are at the heart.
of what makes life possible.
It's pretty amazing when you think about it.
The simple act of a cell dividing.
Is the basis for everything.
Growth,
Reproduction, even evolution.
and the more we learn
the more possibilities open up.
for treating diseases,
Regenerating tissues.
maybe even extending lifespan.
The future of cell biology is bright.
I can't wait to see what we discover.
Me neither.
This deep dive has been fantastic.
I'm glad you enjoyed it.
It's been both fascinating and inspiring.
It's a fascinating topic
I hope our listeners feel the same way.
about the wonders
Of the cellular world.
that drives the complexity of life.
Thanks for joining us on this journey.
It's been a pleasure.